All right, so we're going to talk about DNA replication. The last month or so, right, we've been talking about mitosis, meiosis, right, how chromosomes have to be replicated. What does replicated mean again?
Copy.
Make a copy, right? So we have to make a copy of the chromosomes, of the DNA, so that when the daughter cells are produced, right, they have the same genetic information, correct?
Mm-hmm.
All right, so today we're going to talk about how DNA is replicated, the process, the steps that have to take place for it to be replicated. As we've talked about biology from August all the way until now, right, we've talked about different topics and stuff like that, right, and they all connect at different points, different times. Do we remember talking about enzymes?
Mm-hmm.
Right? What does an enzyme do? Does anybody remember?
It helps make the helps process things faster.
Correct. Yes, it helps the chemical reactions. Yes, it helps chemical reactions to be able to move faster or to go faster. Right? They're known as catalysts. All right. So we have different enzymes that are used during this process, during this DNA replication process. All right. The two enzymes that we're going to focus on today are helicase and polymerase. All right. Remember, we said that whenever we have a word and it ends in ASE, majority of the time it's going to be an enzyme, all right, or some form of catalyst that helps speed up different chemical reactions. All right, so on your paper, we have video recap questions, okay? We are going to watch a video. You guys are going to then answer questions based off of the video. It's all going to have to do with DNA replication. All right, there are 4 different enzymes listed on your paper, okay? We'll answer all 4, but you only really will have to remember 2 of them, all right, that are going to be used in the process. The other 2 we don't really talk about as much, all right? Not going to say that they're not important, but we just don't really talk about them as far as testing and stuff like that goes. You'll probably only see 2 on questions and different test questions and stuff like that, all right? So real quick, let's go through the learning target. So once again, we're talking about DNA replication. Standard is construct an explanation of how the structures of DNA and RNA lead to the expression of information within the cell via the process of replication, transcription, and translation. So DNA replication is a process, like I said, right? There are different steps that take place for this process to happen. All right, we have the replication process, then we have the transcription process, and we have translation. These 2 we'll talk about more next week. All right, we'll get into that because they all have different things that go along with it. Right now, today, we will only really focus on the replication part. All right. The learning objective. Given diagrams and models of DNA and RNA, students will construct an explanation describing how the structures of DNA and RNA contribute to the expression of genetic information through replication, transcription, and translation with at least 80% accuracy on a teacher-created assessment or class activity. All right. Cesar, read the I can statement.
I can explain how the structure of DNA and RNA help explain genetic information in the cell through the processes of replication, transcription, and translation, using accurate vocabulary and correct details.
Good job. All right, so that is the I can statement. So today you guys should be able to explain to me how the DNA replication process takes place, right? So we should be able to tell how the DNA unwinds, right, by the enzyme helicase. Then new nucleotides will be added to the original DNA strand by the help of the polymerase enzyme, right? And then from that, we will get the complementary strands, and together they will both form a semi-conservative DNA strand. Semi-conservative means that one original strand and one complementary strand come together to form a new DNA strand, right? sounds like gibberish right now, like y'all don't really understand what I'm talking about, but it'll make more sense as we go on through the class, all right? But basically, you guys should be able to tell me or explain to me how this process and how the steps and everything take place. So we will watch the Amoeba Sister video. As we watch the video, listen out for different questions that come up, all right? So real quick, let's talk about the questions and what you guys should be listening for as we watch the video. All right, so first question. We've kind of already talked about this in prior topics when we talked about mitosis and meiosis, right? Why does DNA need to replicate before cells divide? Why does DNA have to replicate before cells divide? All right, so this is a question that will be answered in the video. Listen out for it. If you hear it, try to write something down about it so that when we talk about the questions at the end of the video, you guys have an idea of what the answer is. All right. Number 2, it says in the diagram below, label one of each of the bolded terms. So we have to be able to label the different parts of a double helix. All right, so for number 2, we have the deoxyribose sugar. Number 3, we have the nitrogenous base. Number 4, we have the hydrogen bond. 5, we have the phosphate. And then 6, we have the 5 to 3 strand and the 3 to 5 strand. All right, once again, as you watch the video, just pay attention. If you see a part of the video that talks about any one of those 5 different things, try to write something down, try to label a part of it on the diagram, right? Draw a little arrow, write in what it is, so that when we talk about it, you guys are able to answer the question. Number 3, we have the 4 different enzymes. So we have helicase, DNA polymerase, primase, and ligase. All right, these are all enzymes that play a part in the DNA replication process. All right, so they will go through the video and explain each one. Whatever you hear, whatever you remember, write something down about it. All right, and then at the end, we will go through and make sure that everybody has all the correct answers. On the back, we have 4 boxes. All right, we have 4 boxes. They all have a picture, right, associated with them. They are each depicting a part of the DNA replication process. All right, so the 4 boxes show the 4 enzymes performing some type of job or function. Our job is to go in and describe or tell what it is their job is and what they are doing in that process. All right, so we'll go through at the end and we will write in what each one of the enzymes' job is. If you see it in the video, write it down, okay? Understand? Everybody with me? All right.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. And remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has 2 strands, you could think of helicase unzipping the 2 strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and descriptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase, once again, is building a new strand in the 5' to 3' direction. But there is a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules. from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors? In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA basis, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving.
Alright, so we'll stop right there and we will begin to answer the different questions based off of the video. Alright, so first question is to understand DNA replication. So to understand this whole process of why this is happening, why DNA replicates. Alright, why does DNA need to replicate before cells divide? I think Larry said it earlier before we started the video. Why does DNA have to replicate before cells divide? Why does DNA have to replicate? What you say, Caesar?
So a sister cell can have
Not sisters,
daughter,
daughter cells, there you go. So daughter cells can do what?
can have a copy of it.
Can have what? Oh, a copy, yes. A copy, I thought you said pop, I don't know what I was hearing. But yes, a copy. Alright, so to make an identical copy of DNA, so each new cell has the same genetic information. I'll make this a little bigger so that y'all can see it. Can y'all see that? Alright, on the next part, in this next box over here, we have to label the diagram. Alright, so we have to label the double helix. Alright, so whenever we see like a drawing of a double helix, it normally looks like something like this, correct? We've all seen this in like a movie or a video game, right? We've seen this double helix structure. We always see these different lines that go in between that look like a ladder, correct? Alright. This is really what it looks like, okay? We have a deoxyribose sugar, and then in the middle, this latter part, right, that we see, are really nitrogenous bases, okay? So what we need to remember is when we are talking about DNA, we have two pairs that we have got to remember, all right? Whenever we see a question and it's asking us to draw a base pair or to draw a double helix or whatever, right? If we are putting in pairs, we have to remember that A equals T, T equals A. This is going to be important when we start going through transcription and translation. All right, you'll have a DNA sequence where we'll have a list of different pairs, right? And you'll only be given one strand, and you have to go in and fill in its pairs based on the different rules. Does that make sense? All right, so in a second when I give you your practice, there will be a question where you'll have an original strand and you have to make the complementary strand, right? So basically we'll take off this red side here and you have to go in and fill in its base pairs. So you have to be able to remember A equals T, G equals C. Does that make sense? Then when we get to transcription and translation and we start dealing with RNA, the base pairs will change just a little bit because A will then equal U, all right? but don't worry about RNA right now. We'll get into that part a little bit later. So in the middle here, we have the nitrogenous bases. Once again, A equals T, C equals G. They always are paired together. Okay? So for the first one, we're just labeling. All right, so all you're doing is just taking an arrow, drawing a little arrow wherever the different parts are, the motor terms are. So this first one, deoxyribose sugar, is going to be this yellow shape right here. All right, so we should draw an arrow from that little part. So on your paper, from any one of them, draw an arrow and label it deoxyribose sugar. How many do we have in this diagram? Who? 6, good. On number 3, it says nitrogenous base. Nitrogenous base. These 6, are all nitrogenous bases. All right, they each have a name too. So A is adenine, T is thymine, C is cytosine, G is guanine. All right, they all have a name that go along with them. How many do we have? 6. So we should have 6 written up here as well. I'll bet I'm on your way. So for number 3, we should have an arrow pointing at one of these 6 nitrogenous bases, have an arrow pointing, and then just label it nitrogenous base. Move this over to the side. All right. Giuliani, you all right? You still got a headache? No? 4 is the hydrogen bond. Hydrogen bond is going to be in the middle. Okay? So they bond the 2 bases together. So it's bonding adenine and thymine together in the middle. Okay? Just a little bit of extra information if we ever, I don't know if we'll ever see this on a question, but the bond that is over here that connects the phosphates to the deoxyribose sugar, they are called covalent. So the sides of your ladder, right? So if you think about a ladder, right? Everybody's been to like a construction work site or seen a construction work site or seen a ladder or something like that. The sides of the ladder is going to be covalent bonds going up. All right. The middle where you step to walk up is going to be your hydrogen bonds. Okay. Hydrogen is the stairs. Covalent is the sides where you hold on to. So hydrogen bond is going to be in the middle. All right, how many do we see?
3.
3. And then for number 5, we have phosphate. Phosphate is going to be over here. So they're going to have this little circle. Okay, that is your phosphate. So you draw an arrow coming off and just label it phosphate.
What is it?
Right here, phosphate. So choose any one of the circles that you see on your diagram and write it in. All right, and then number 6, we have the 5' to 3' strands. All right, the way that we are able to visualize and know which is which is we are counting from your deoxyribose sugar. All right, if it is not already labeled for you, all you have to do is count. We always start at the top, the triangle part, the part that looks like the top of a house. Okay, we start here, that is 0. and we go clockwise. So think of a clock, think of where 1 is, and we go 1, 2, 3, 4, 5 is always going to be this bond that comes out of the house. So think of like your chimney or something like that at the top of a house. Okay, that is your 5. So if your 5 starts here, this is 5 to 3. If we look down here at the bottom, we see the 3, correct? It's at the bottom. Here, what's at the top? 3. So this is going to be your 3 to 5. Okay? All right? So that is how you are able to determine excuse me, which is which. All right? That's how you're able to determine. So your leading is going to be 3 to 5. Lagging is going to be 5 to 3. So you should have labeled on this side. You could write it along the side if you want to. Lagging over here. Lagging over here. Leading over here. Your leading strand is always going to be your original. So your original strand is always going to be your leading one. Okay? So when the polymerase goes in and is adding nucleotides, right? This side over here, right, we won't have any type of lag. Remember on the video they were talking about a lag, and we had to have ligase come in and connect the different fragments, right? So on your 3 to 5, there will be no lagging, right? 5 to 3 is where you will have the lag. That's why it's called the lagging strand, or lagging side. All right, that's going to be your complementary strand. All right, real quick, since we don't have much time. We will write in the answers here. And then I want to give y'all the practice so that y'all can take it home and y'all can see the, y'all can practice putting in the base pairing rules. You should be able to answer these questions just based off of what we talked about today. So if you look on the back, right, on number 11, you see how you have the original strands, right? And you have to go in and you have to add the complementary strand based on the base pairing rules. Do you see that? Everybody see what I'm talking about? So the first one says A. What will we put for the complementary strand? T. The next one says T. What will we put? Huh? A. G. C.
G.
Good. So you would do that all the way through the strand. You don't understand, Cecil? Cecil looked up like, oh, what is he talking about? So, using your base pairing rules, Cecil, up here. This is DNA, correct? Using your base pairing rules, you have an A written first, correct? Yeah. A equals what?
T. So
on your complementary strand, you would write a T right under it. Does that make sense? T goes with A, so you write an A right under that. G goes with C, so you write a C right under it, so on and so forth. Does that make sense?
Yeah.
All right. So that is how the complementary strand, that's how polymerase knows what to go, what nucleotides are added, what bases are added for the complementary strand. So we can have 2 identical DNA strands. All right, so real quick, let's finish this assignment. I just wanted to show you that because I know y'all getting ready to leave soon. So if you have your phone on you, I'm gonna type in these answers real quick, and then you guys can take a picture of it, all right? From this part right here, from number 3, you guys should be able to go to the back side of your worksheet, right? And be able to tell what's going on in the different pictures, what the different enzymes do. Think you got it? Okay? Alright, so real quick, I'm gonna type these answers in. Alright, take a picture of this so that you guys can write down these answers on your notes. Can y'all see it?
Yeah.
Joel, you took a picture of it? Alright, so you guys are typing in this, not typing in, but writing this down, and then using this right here, you guys can fill it in with the different pictures. Alright, does that make sense? So for your homework, I want you guys to go home. These are just going over the same thing for your homework for this weekend.
Yeah.
Go through, answer these different questions. Alright? All of these we should be able to answer. We should be able to answer. Complete the complementary strands. Alright, complete these complementary strands. Alright, and then tell me what does semi-conservative mean. So, semi-conservative. So remember, semi-conservative, one original strand, if you want to take a picture of this as well. One original strand, one complementary strand. So one blue, one red comes together to make a semi-conservative. Alright, or a copy, a DNA copy. Alright, with the information that I gave y'all today, with the notes that I gave you, the video recap questions, alright, you guys should be able to answer all of the questions on your homework. Does that make sense?
Yep.
Yes, Juliana, do we have any questions? So on Monday, not Monday, y'all come back Tuesday. On Tuesday, when y'all come back, we should be able to have this finished, correct? Alright, cool.